Hello, grade 12s. So we're moving on to a section now about DNA replication, as in how am I going to be duplicating my DNA? How do I create exact copies of my DNA? So in the previous video, we looked at what is the generic structure of nucleic acids, but what we're going to be focusing on now is just DNA itself. So we're going to be focusing on the things related to DNA. And where does this actually fit in the rest of the applied nature? Well, DNA replication actually really makes its appearance when we're talking about cell division. So when we talk mitosis and meiosis. So it might seem a little bit out of place that we're putting it here, but it really does help for you to get your understanding of protein synthesis, to have an understanding of how DNA replication takes place and how that complementary pairing works before we move on to going to protein synthesis itself. So, as a recap from the last video, we know that DNA has many nucleotides that are chained together. So essentially DNA is a polynucleotide with many of these nucleotides chained together. So for this particular video, I'm going to use this model to represent a nucleotide. Again, remember, I know that we had a thing previously where you have to have like a 5-sided sugar and the circle and that. Just for this video, I am wanting to use this for ease of representation. So what you're seeing here is that the light pink rectangle is going to represent the sugar. In this case, the DNA sugar, which is deoxyribose. The little darker purple rectangle will be the phosphate, and the blue with the letter will represent the nitrogenous base. So as a reminder, you have adenine, thymine, cytosine, and guanine. So the letter at the beginning is just what I'm going to use to represent each different base. So this is my model of a nucleotide. So let's give you a theoretical example. Here is a DNA sequence. So just a theoretical DNA sequence of letters. I want you to, as a little exercise to see if you remember what happened from the previous video, what is the complementary DNA sequence of the one that is given to you here? Pause the video, write down all these letters, write down the complementary pair for yourself. Okay, I hope you did actually practice that, because you do need to have an understanding of complementary pairing. But starting from the bottom, going towards the top, I hope that you got this sequence of letters. So check that you got these correct. So a reminder, if you have adenine on one side, you'll have thymine on the other side. If it's thymine on one side, it will be adenine on the other side. Cytosine on one side will be guanine, and guanine on the one side will be cytosine. You have to have an understanding of this complementary pairing. So it's not exact. Okay, so that is how I can complementary pair to create one strand of DNA. So, we know that when we have DNA, we know that it is a double helix. What does that mean when we have a double helix? Well, if you see here, this pink bit is now representing the sugar phosphate backbone. So the two pink bits, sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. That is staying the same down the molecule. What is then being different is what's happening in the middle with the nitrogenous bases. So remember, we have the nitrogenous base on one side, a hydrogen bond linking the nitrogenous base on the other side. And how are they linked together on each side? In complementary fashion. That's what we mean. Okay. So this is what a normal DNA molecule is. So what we are concerned about in our video is DNA replication. So what do I mean is I'm looking for when a cell divides, as in mitosis or meiosis, the DNA must be copied, as in it must be exactly the same as the original for when it's going to move into for the new cell as what it was as the parent cell or the haploid cells for meiosis. And what do I mean that it needs to be copied exactly? What I specifically refer to is the order of nitrogenous bases that are occurring. So the order has to stay the same, because remember, DNA is the code of life. If I start switching around those codes, changing places of things, I'm going to cause havoc in my cell. So I need to make sure that those are copied exactly as the original was. So if I copy it across over, these two should read exactly as the letters go down this way, as these letters go down this way. So how do we do it? So here's just a very, very brief overview before I go in detail. So what happens in DNA replication is we have to separate the DNA first. So the first enzyme moves through. So this little green heart represents a particular enzyme. I'll get to that in a second. And what happens is it breaks the hydrogen bonds linking the two chains together. So it literally goes down the middle of the two polynucleotides and it breaks those hydrogen bonds. And then those two chains separate. So I have one polynucleotide chain here and one polynucleotide chain here. I still have the nitrogenous bases here. Don't think that they've gone away. The nitrogenous bases are still here, adenine, thymine, all that kind of stuff, and the same on this side. But I've now just separated the two of them. Then what happens is I have free-floating nucleotides, a sugar phosphate and a nitrogenous base, connected, individually floating around. They then come in and in a complementary fashion will form the new strand on either side. And in that way, I have then created two identical strands of DNA. So let's see how that works a little bit more in depth. So here's a closer look. Here's my DNA strand that I gave you guys here. This is the letters that I had originally. So the enzyme that breaks it is DNA helicase. So DNA helicase, again, the helicase sounds like the word helix. Remember, the shape is a helix. When we use an ase, the suffix for an enzyme, it's like a breaking down kind of thing. We're not making it, we are breaking it down. And we're specifically relating this to DNA, the structure of DNA. Yes, you need to know this enzyme. Please know it. So again, DNA helicase, it comes and it breaks the hydrogen bonds in the middle. The two strands then separate. Now, another enzyme called DNA polymerase links free nucleotides to form the chains that are identical. So, DNA polymerase. What is polymerase? Poly meaning many. Polymerase is basically to make more of. That's what polymerase is. Whatever the polymerase, the word that happens before it, polymerase is making more of it. So in this instance, DNA polymerase is making more DNA. There are other types of polymerases out there. So just remember, in DNA replication, I am looking at DNA polymerase to make DNA strands. And I take my free nucleotides, free floating ones, and I'm going to link these in complementary fashion. So you're going to do this for a second for me. I want you to write down these two sequences with a space between them, and then I want you to practice. Write down the complementary DNA strand on this side, and I want you to write down the complementary DNA strand for this side. Pause the video and do both of those now, please. Yeah, so I hope that you've done that. Really get into the practice of knowing what this complementary stuff is. So let's check. Did you get it right? For the left-hand side, did you get the following order? Check your answer. Pause if you need to. And for the other side, did you get the following order? Check your answer. Pause if you need to. Now, if you were clever, you would have noticed that the letters on this side are exactly the same as the letters on this side. And the letters on this side are exactly the same as these purple letters. So yes, exactly, complementary fashion. Now if I look at these two DNA strands, I now have exactly copied my DNA. And if I did this for the whole structure, it would therefore be exactly the same. And that is basically DNA replication. I separate the bands with DNA helicase, they separate, complementary fashion, I bring free nucleotides in one at a time, link them in complementary fashion, and I have then created my new DNA strands. Simple as that. So, please go back, review, understand which enzymes do what. You need to know those enzymes. Review complementary base pairing. It becomes very important when we go into protein synthesis. You have to have a strong understanding of that. And otherwise, just make sure you have a general understanding of this for when we go into meiosis at a little bit of a later stage. If you're struggling, hit me up on Skype, get me on Teams, let's have a meeting, and we can discuss these a little bit further. Otherwise, thanks very much, guys. Hope you enjoyed that video.